Welcome to Study Loop.
Alrighty, so problem 44 here is a long one, so we'll dive right in. And the first thing it wants us to do is describe the differences in each blood cell type, right? So if we have our four different types of red blood cells here, there are antigens, right, that phenotypically get expressed on the surface of the cell, okay? And so we have our A antigens. We also have a B antigen. Um, we could have both antigens, or we could have neither antigen, right? And so if we are expressing phenotypically the A antigen, then that's our A blood type. And if we're expressing the B, that's our B blood type. If we're expressing both, that's where we get the AB blood type, and neither gives us the O blood type. Okay, so this one is part A. So moving on to part B, this is about how the blood type group does not display the Mendelian inheritance. Okay, and so what's meant with that is that with Mendel, if you have two genotypes, right? So if you'd have like big A, little a, and like big A, little a, right? You could do your little square here and see that there's a dominant one, a recessive one, and three of them would express whatever your big A trait is, and one would express your whatever your little a trait is, okay? But with blood cell types, so if we had a type A and a type B, and a type A and a type B blood type for alleles, okay, you'll get your one person with a type A blood type and one person with a type B blood type, but then you'll get two of these with the type AB blood type, right? So instead of one of them being dominant over the other, both get expressed. Okay, and so that's where that deviates from. And so this idea is part B here. And moving on to part C, here we're given a little story about how our T cells, okay, they recognize these elements on the surface, and they recognize them in both bacteria or our red blood cells, right? So they can actually sort of recognize these little antigens. And this is why if you put the wrong type of blood into someone, the T cell will recognize a foreign antigen and sort of attack it, right? So you can't really use incorrect blood or non-matching blood, I guess, would be a better way to say that. And so we're told that the T cell can sort of recognize these components on both bacteria and red blood cells. Okay, and then it further asks us, well, first it also tells us that certain blood types, right, are more susceptible to various infections. So we're told that our O blood type is susceptible to cholera, okay, and that our AB blood type has shown increased susceptibility to E. coli. And then it asks us, well, what is a possible reason for this, right? So we're just sort of brainstorming why this might be. Well, if our T cells are able to recognize these sort of surface elements, then it's possible that as a E. coli, if we let this be our E. coli bacteria, and say we had a AB blood type here, there's might be something on the surface of the E. coli that looks similar to our A or similar to our B antigens that's stopping it from releasing the necessary antibodies that would kill it, right? And so what kind of data would we need then to test this reasoning? Well, one thing would be to also investigate the susceptibility of type A or B against E. coli, right? Because if that is the case, where if it's just one of them, then that other blood type may also show increased susceptibility. Okay, but it could be something else, right? Maybe there's something, maybe it expresses something else entirely that just sort of resembles the combination of the two, right? So maybe some sort of molecular level investigation into those carbohydrates would be a better selection of data. Okay, so that was part 3, is sort of just a little thinking exercise, and then we get to 4 here, and it starts to get a little bit mathy, right? Which is great. So we're given observed number of folks with a blood type, which is in this column, and the total, right, which is at the bottom. And we're just asked to find the frequency, which is not too hard, right? So we just need the total number observed. So for blood type A, it'll be 501. And you divide it by the total number of people. So that'd be by 2132. Okay, and this would give us a frequency of about 24%. We can go ahead and put that in there. Okay, then we just sort of repeat the process, right? So for blood type B, it would be 794 divided by the totals. That's 2,132, and then that one's going to equal 0.37, and so we can put that in there. And I think you sort of get the hang of how to figure that out. So the other, for AB, it'll be 0.11, and for type O, it's going to be 0.28. Okay, and so then we sort of get going on, and someone has gone in and used this data to give us some probabilities. Okay, and so they give us that the probability of allele A is, for blood type A, is going to be equal to 0.201. For B, it is equal to 0.265, and this information is in the problem. And for probability for O is equal to 0.542. Okay, and then we're asked to calculate some of the expected frequencies, and they give us a equation, and 2 of these equations have a typo, which makes it incredibly confusing. Okay, so the expected number of people we would expect to have this blood type A is going to be equal to the probability of someone having the allele A squared plus 2 times the probability of A times the probability of O times the total population. Okay, and this is because the way in which you get a type A allele is if 2 A alleles come together, or an A and an O allele come together, right? And so that's what that represents. And we get 2 here because you could get an A and an O, or an O and then an A, right? And both of those will have the same result. Okay, so if we just punch in our numbers for this, then we would get that 0.201 squared plus 2 times 0.201 times 0.542, and multiply that by the total, and that total n was from that previous table of 2,132. And once you type this into a calculator, all right, we find that this is going to be equal to 550. Then we can just put that in our graph. And now part B is done the exact same way, right? Except everywhere you see 0.201, we would write 0.265, right? And same with here, 265, and then the resulting number is going to be 762. Okay, then AB had a little bit of a different equation to it. Okay, so that one is just, so the expected value for the AB, that one is equal to 2 times the probability of A times the probability of B times N. So that is 2 times 0.201 times 0.265 times 2,132. Right, and that number comes out to 227. And then the probability of the expected number of folks we would expect to have O then is, I'm going to tuck it in the corner because it's a short equation here. So expected value for O is a nice one. It's the probability of O squared times N. So that's just the 0.542 squared times 2,132. Okay, and that number ends up being 626. Okay, and so once you have the correct formulas, it becomes way easier. And this is still part D, both these two screens were, and this next one is also going to be part D. This one's asking us to do a chi-squared test, and the chi-squared formula says we want to take the sum of our observed value minus the expected. I should get rid of that really big parentheses. We just need a little one. So the observed value minus the expected squared divided by the expected. We do this for one less variable, right? So if we look at this previous page, we see we have 4 total variables. Okay, so we just have to pick 3 of them, right? So I'm just going to pick, for simplicity's sake, the sort of top 3 here for A, B, and AB. But you could do any 3. Okay, and so this is going to give us then a pretty long formula, but not too complicated. So our observed was 501, and we calculated our expected to be 550. Square that and divide it by the expected, so it's by 550. Right, and we do the same thing for all of them. So then we get 794 observed minus the 762. Square that and divide by 794. Okay, and then we have one more piece to add on. So we'll add on the 236 minus 227. This is from our AB. Square it, divide by 236. Okay, and if we calculate all that out, we end up with 6. Okay, and so then we just need to go and compare this to our chart, right? So we were looking at the 95th percentile for 3 variables, or 3 degrees of freedom, right? So the degrees of freedom are here, 3, and with the 95th percentile, so where that lines up is 7.82, and we had a 6. And of course, when I did this, I did a little bit of rounding, so this, you might get a slightly different answer. But that's pretty dang close, right? So that would suggest to me then that this sort of supports the idea of the Heidi-Weinberg equilibrium. Right, so if something else was going on, then that number would be, our number would be quite different from this one. Okay, but instead, we're finding that the chi-square test sort of suggests that this particular population is not seeing a change in the allele distribution, right? And so since we're not seeing a change in allele distribution, what kind of data would we need to further test it? Well, it's all about there not being alleles over time. So what you'd want is data from a future time that you could compare it to. Okay, and then that leaves us just with this sort of last little chunk, part E, finally. And this time we're told that there's homologous genes coding on that are present on the surfaces of red blood cells, right? And this is in both amphibians and mammals, but not fish. Right, so we have these red blood cells, and we have these things being presented on them in mammals and amphibians, but not fish. So we have in mammals and amphibians, but not in fish. Okay. And so this sort of implies that the common ancestor to these ABO genes, these things are being presented, right, occurred at least 20 million years ago, somewhere around the time when we got a distinction, separation from fish and amphibians. And so the difference between genes coding actually for A or B is a single nucleotide. So if you're A, C, T, G, etc., and then maybe the difference is just something like that. There's one single nucleotide that separates you from being a type A allele or a type B allele. And so what is the likelihood that negative selection pressures have been active in the evolution of this system? Well, the evolution of this system, okay, well, first, negative selection is the removal of a undesirable trait, right? So it's basically Darwinism, as like selection of the fittest. So if we had negative selection of the whole ABO system, you would expect to see fish sort of die off, right? Because they don't have it. But that's not the case. There are plenty of fish, and fish and mammals even sort of coexist in the same habitat, right? So I would argue that there is not negative selection for the ABO system. But if we are investigating just A and B blood types, then we would need perhaps a little bit more data. Okay, so with the AB blood types, we would want to look at the frequencies of the A type versus the B type over time. And if you see that there was a time when one didn't exist, right? So if for some period of time we only had type A, and then B popped up in a small value and then became more and more prevalent, that might suggest that there is some selection. But if they've both always been present and always without too different of a degree of frequency, then it would be likely that this negative selection pressure is again